Hi guys, I'm Antonio Macedo. I'm a senior TA candidate for this year's program, and the topic I chose to talk about is clinical research. What does history tell us? As a general outline of my presentation, I'm going to talk about the definition of clinical research, the scope of clinical research, some of the ethical issues involved in the history of clinical research, as well as some of its unique issues and particularities, some of the regulatory aspects and norms, and we're going to end this session with a brainstorming session where we'll have some questions to try to incite some discussion among the students. And of course, I'm going to include the references. So what is clinical research? We know that the search for knowledge about ourselves and the world around us has driven humanity ever since its existence. So research as a whole is actually derived from this desire and curiosity to understand and improve the world. The scope is thus gigantic. So it basically includes four levels. Physical level, which has to do with the study of the origins of the universe, the nature of matter, and so on. The analytical level, which includes maths, logic, metaphysics, and so on. The basic or preclinical level, which includes basic science, animal studies, micro studies with microorganisms, and so on. And of course, the level which has more to do with what we're going to talk about, which is clinical level. And of course, it includes human beings. Um, the clinical level studies include both the history of humankind, the human body, social, cultural aspects, political ones, philosophical aspects, and so on, not just the biological ones. So it is actually boundless. Carl Sagan, a famous astronomer, has a saying that goes, somewhere something incredible is waiting to be known. So this really has to do with what research is about. It's about the search for the unknown. An interesting definition of what clinical research is comes from our program book, Professor Fregni and Dr. Illigan's book, which states that clinical research is the branch of medical science that determines the safety and effectiveness of medications, devices, diagnostic products, nutrition or behavioral changes, and treatment regimens intended for human use. Another interesting definition comes from Zora Neale Hurston, who was an American anthropologist, and she stated that research is formalized curiosity. It is poking and prying with a purpose. So, going a little bit deeper into the topic of clinical research and some of its core principles, we know that clinical research is a structured process of investigating facts and theories and exploring connections, and it can have several goals. And it proceeds in a systematic way to examine clinical conditions and outcomes, to establish relationships among clinical phenomena. It can provide the impetus for improving methods of practice, and of course, it can generate evidence for decision making. So basically, clinical research comprises a set of procedures in medical research and drug development that are conducted to allow safety and efficacy data to be collected for health interventions. And these interventions can be drug therapy or other kinds of therapies, and procedures. Still, regarding some of the core principles of clinical research, it can also be defined as simply as experiments conducted in humans, which, as I said, comprises a series of required procedures and steps to conduct clinical trials. And the overall purpose of a clinical trial is to acquire knowledge that is novel, not the actual treatment of patients per se. So we're going to talk about clinical trials, which can also be defined as patient-oriented research. So what is patient-oriented research, or what are clinical trials? They actually comprise any investigation involving participants that evaluates the effects of one or more health-related interventions on health outcomes. So it's basically my biomedical research, but it can also include other related disciplines such as psychology and even the social sciences. And it can include various amounts of interventions and that have been listed here, and they are not restricted to this list, but these comprise the main ones: drugs, radiopharmaceuticals, cells and other biological products. In other words, cell therapy that can include CAR T cells, donor lymphocyte infusions, hematopoietic stem cell transplantation, and so on. Surgical procedures, radiological procedures, devices, genetic therapies, even natural health products and dietetic or behavioral interventions, process of care changes, preventive care, manual therapies, or other kinds of alternative therapies, alternative medicine therapies, psychotherapy, and so on. These are all types of interventions that can be used in clinical trials. And these trials may also focus on metabolic pathways and mechanisms of disease that are not really directly treatment related. Anyhow, most of the time, clinical trials basically compare two or more approaches in regard to a given outcome. So who conducts clinical research? Basically, the researcher leading a clinical trial is generally a clinician, but he can also be any healthcare provider. And there are various types and forms of clinical trials with methodological differences, but the ethical principles behind them and the procedures that they comprise are virtually the same and applicable to all. So going a little bit into the history of biomedical experimentation, it actually dates back to almost 5,000 years ago, to the year 2737 before the Common Era. And one of perhaps the first clinical trial that was ever documented is in the Old Testament. I'm going to talk about it a little bit in a couple of minutes. And from that time onwards and until today, of course, clinical research has changed and progressed immensely. There has been an enormous refinement of regulatory norms, methodology, scope, and practice regarding research, and especially in the last 50 years, as I'm going to show you in the next couple of slides. So starting off with some milestones related to clinical research. In the year 2737 before the Common Era, that is around 5,000 years ago, Shen Nung, who was a legendary emperor of China, and is considered the father of Chinese medicine, including of acupuncture, experimented with and classified hundreds of poisonous and medicinal herbs in a series of studies which he actually conducted on himself. Some 2,000 years after that, in the year around 600 before the Common Era, the Old Testament, as I had mentioned, mentions the first trial, which we can today consider a pilot study, in which under King Nebuchadnezzar II, children of royal blood and certain children from conquered Israel were divided into two groups. The first group would receive the king's diet, which was basically comprised of meat and wine, versus the second group in which the children would receive restricted diet, basically legumes and water. And these groups would be followed for a 10-day trial period and then assessed for outcome. And they actually found that superior wisdom and understanding was seen in the restricted diet group, not in the king's diet one. Some 1,000 years after that, or 2,000 years after that, in 1537, a French surgeon named Ambroise Paré accidentally discovered that a mixture of egg yolk, turpentine oil, a kind of oil, and rose oil was efficacious in the healing and relief of pain of gun wounds. This was compared to elderberry oil, which was back then considered the standard treatment. Still regarding some interesting milestones that we have in the history of research, In 1747, the first reported systematic experiment of the modern era was actually performed by Dr. James Lind, a Scottish physician, while sailing on the Salisbury. Salisbury was a British naval ship, and Dr. Lind, for this study, selected 12 what he called similar sailors with scurvy. And these sailors were split into 6 groups of 2. All of them received the same diet, but group 1 would receive cider, group 2 would receive vitriol, which was sulfuric acid, group 3 would receive vinegar, group 4 seawater, group 5 oranges and lemon, and group 6 would receive nutmeg and barley water, which was a kind of British herbal tea. And the outcome that Dr. Lind evaluated was recovery from scurvy. And this was, of course, based on a clinical type of assessment. And group 5, which comprised oranges and lemon, was the one which recovered the most quickly and in just 6 days. And among the other 5 groups, group 3, which received vinegar, showed the second best effect. So after that, around 5 or 6 years after the study, Dr. James Lind published his Treatise of the Scurvy in 1753, which didn't really have a short-term impact because there were several challenges back then, especially regarding the preservation of citrus fruits, as well as the expense related to preserving such fruits. Anyhow, about 40 years after that, the Royal Navy turned fresh lemons into a standard sailor supplement. So we can already make out from this study that some clinical research was already starting to be performed, and that was already managing to change practice a little. In 1932, almost 150 years after the original study, finally some solid evidence of the link between vitamin C and scurvy was shown. Still regarding some important milestones, in 1863, Dr. Austin Flint, a US physician who graduated from Harvard Medical School in 1833, conducted the first study using placebo, or what he called placeboic remedy, which was a substance or procedure with no presumed therapeutic effect. And he studied hospital inmates who had rheumatic fever. And these hospital inmates, while receiving placebo, were compared with patients who had received an active treatment. But there were many drawbacks of this study, and basically its ethical aspects were questioned because Dr. Austin Flint was studying a vulnerable population comprising inmates or prisoners, and of course there was no consent. Regarding the active treatment that he used, it seemed that it wasn't so active, or perhaps had questionable efficacy to treat rheumatic fever. And another point was that the groups were not compared or not tested at the same time. So in a way, these placebo was compared to active treatment, but not concurrently. So still regarding some of the milestones, now some more recent ones in the last century, 20th century. In 1923, the idea of randomization was finally introduced to some studies. And of course, as we know, randomization allowed for the fact that participants would be randomly assigned to one of the treatments, let's say a new drug versus placebo. And some 20 years after that, the first double-blind control trial, which used patulin for the common cold, was conducted. Just a year after that, the first clinical trial, multi-center clinical trial was introduced, and it actually comprised multiple trials that were conducted at various sites at the same time and using the same protocol. This allowed for wider testing, greater generalizability, and of course, acquisition of more robust statistical data. Some 4 years after that, already after the Second World War, the first widely published randomized control trial was conducted on streptomycin as a treatment for pulmonary tuberculosis. And this randomized control trial was published in the British Medical Journal. So I'm going to talk a little bit about the history of ethics in clinical research, which we will note that was originally an utmost disaster. So perhaps because of that, clinical research nowadays is perhaps the most regulated type of research, of course, because it involves human beings, life. And we know that a significant part of the existing ethical regulations and norms in clinical research that we know of today were fostered by a myriad of disastrous events that took place at the beginning of the new era of clinical research, basically the 20th century onwards, particularly after World War II, when experimentation and the development of novel treatments began to take place in a more systematic way. So all of the changes that we've witnessed during the last 70 years or so, both in the US and globally, were actually fostered by human catastrophes, which eventually led gradually and progressively to ethical regulations of human research. And despite some regional differences between these ethical regulations, they all took a similar pathway worldwide. So regarding the beginning of such regulatory norms, in 1906, around 120 years ago, the Pure Food and Drug Act imposed purity standards on products and drugs and mandated accurate labeling with content and dose. So although we take this for granted nowadays, it was only like 120 years ago that this started to be performed. An interesting study, or not study, sorry, but situation which was witnessed in 1937 was that of sulfanilamide cold syrup that was used for streptococcal infections. So it was made of an elixir dissolved into ethylene glycol that we know is an antifreeze toxic substance. And this formulation was used instead of an already available formulation, which was in tablet form or powder form. However, this syrup form had not been tested for safety at the time, and this unfortunately led to lethal effects in hundreds of people who had the common cold or throat infection. They died 10 to 21 days after taking the medication. And this was an absolute disaster. So as a result, just 1 year after that, 1938, the Federal Food, Drug and Cosmetic Act, the FDFDC Act, also from the US, required that new drugs be shown to be safe before marketing and that safety limits be set for unavoidable poisonous substances. And this, of course, meant a stricter control of the production and distribution of new drugs, and it also stimulated medical research. Another intriguing and really sad event that we have regarding clinical research has to do with the Tuskegee Study, which took place from 1932 to 1972, so around 40 years, and it was named the Tuskegee Study of Untreated Syphilis in the Negro Male, and it was actually performed in a poor rural area of Alabama in the US, and it was meant to assess the natural history of the disease. And the sad thing about it is that it was actually sponsored by the United States Public Health Service, as well as the Tuskegee Institute. In this study, hundreds of African Americans were left without already available treatment, since penicillin had already been discovered in 1928 by Dr. Alexander Fleming. And these African Americans that were left without treatment, eventually many of them died or had their descendants be born with congenital syphilis. And another sad thing about this study was that it was not ever kept secret, and there were dozens of publications regarding this study. And the main justification for the continuation of this study for so many decades was that it contributed to science. And this contribution to science by far outweighed its detrimental effects on the health of the study population. So another important point is that the Tuskegee trial continued irrespective of the Nuremberg Code, which was set in 1947, and even the Declaration of Helsinki in 1974, as well as the opposition of the Catholic Church that urged physicians and scientists to always respect patients, superseding all scientific or research objectives. These are just some images from this cruel study. Another important period in time regarding clinical research and human experimentation had to do with the Nazi regime, and this, of course, took place during the Second World War, from 1939 and 1945. And this occurred amid Nazi Germany. And this political structure, which is based on a totalitarian system, also included the so-called racial hygiene paradigm, and also there practically inexisted any legal or ethical boundaries for human experimentation back then, at least in Germany. So this provided the ideal conditions for inhuman experimentation. And sadly, unimaginable cruelty and disrespect for unconsenting subjects took place, with an absolute disregard for human suffering and for human life. Still, in regard to the Nazi regime and the experiments that they conducted, it's important to note that some of these studies were actually conducted according to the scientific progress of the time, whereas others were using obsolete or outdated practices. In any circumstance, horrendous experiments were performed and thousands of deaths were seen. And still of note is that there is no direct evidence that any of the researchers involved were ever forced to participate, nor that any of the research techniques and practices were actually directly imposed by the government. And the main justification for this was actually that these studies were performed under the ideal of preserving the health and well-being of the population, at the same time that new critical knowledge was being gained from such experiments. So these cruel experiments finally led, a couple of years after the end of World War II, to the promulgation of the Nuremberg Code, which included 10 basic statements for the protection of clinical trial participants, following the judgment of Nazi physicians in Nuremberg, which is known as the Nuremberg trial or the Nuremberg trials and the doctor's trial, which was the precipitating event for the start of modern research ethics. This is just an outline of the Nuremberg Code. I'm not going to read all of it for the sake of time, but anyhow, I'm just going to point out a few highlights here. So the Nuremberg Code comprised 10 statements, as I mentioned, and the first of which had to do with the voluntary consent of the human subject. The second, the experiment should be such as to yield fruitful results for the good of society. The experiment should be so designed and based on the results of animal experimentation and a knowledge of the natural history of the disease or other problem under study that the anticipated results will justify the performance of the experiment. The experiment should be so conducted as to avoid all unnecessary physical and mental suffering and injury. No experiment should be conducted where there is an a priori reason to believe that death or disabling injury will occur. The degree of risk to be taken should never exceed that determined by the humanitarian importance of the problem to be solved. Proper preparations should be made and adequate facilities provided to protect the experimental subject against even remote possibilities of injury, disability, or death. The experiment should be conducted only by scientifically qualified persons. During the course of the experiment, the human subject should be at liberty to bring the experiment to an end if he has reached reached the physical or mental state where continuation of the experiment seems to be, to him, to be impossible. And similarly, during the course of the experiment, the scientist in charge must be prepared to terminate the experiment at any stage, if he feels that he should do so. And these are just some images from the Nuremberg trial back then, starting from 1945 onwards. And even so, after this trial was performed and the Nuremberg Code was published, there were some statements that were said by an American politician some couple of decades after that, Paul Neitz, and he said that we slow the progress of science today for all sorts of ethical reasons. Biomedicine could advance much faster if we abolished our rules on human experimentation in clinical trials, as Nazi researchers did. So, despite all the advancements that we witnessed following, especially during the 20th century, unfortunately, these Nazi and unethical ideas still exist in more recent times, sadly so. So, carrying on with a little bit about the regulation regarding clinical research, and another important historical aspect of it has to do with thalidomide, which in 1957, some 4 years after it was first discovered, thalidomide, which was known as an anticonvulsant and was also beginning to be used as a sleeping aid due to its sedative effects and also as an antiemetic, especially for morning sickness in pregnant women, began to be sold like off the shelf, and there was an unregulated and widespread use of it, of course with a lack of well-designed safety studies, and eventually it was finally shown that thalidomide could provoke irreversible peripheral neuropathy in chronic users, and also birth defects and growth retardation. So this situation further exposed the ill-equipped regulatory process at the time. So these are just a couple of pictures where we can see the birth defects. So one of the important milestones regarding the regulation of human research was that of the Kefauver-Harris drug amendments in 1962, which established a tougher price and safety controls on pharmaceutical company products. It also required that evidence of safety and efficacy data be based on well-designed and conducted clinical studies using up-to-date techniques and practices. It also pointed out that all clinical trials would have to be approved by the FDA, all trial participants would have to be properly informed and consented, and the FDA could immediately remove ill-proved drugs from the market. In 1964, some 2 years after this last statement, the Declaration of Helsinki was published and by the World Medical Association, and this declaration outlined a number of ethical codes for physicians and the protection of clinical trial participants worldwide. It did this by highlighting the importance of conducting concise but thorough review of any research protocol that involves human experimentation. So this is just a picture from the World Medical Association site stating the Declaration of Helsinki and its ethical principles for medical research involving human subjects. And there were a number of assemblies, general assemblies, sponsored by the World Medical Association, and even in Brazil last October, in October 2013, which was the 64th WMA General Assembly. So there were some amendments during the last 60 years or so. And the Declaration of Helsinki has a set of general principles which comprise around 37 items or principles. And these 37 principles are comprised within these main topics. So privacy and confidentiality of human subjects, scientific requirements and research protocols, risks, burdens, and benefits for trial participants, vulnerable groups and individuals, the importance of research ethics committees, post-trial provisions, the use of placebo, informed consent, vulnerable groups and individuals, which is actually being repeated here, unproven interventions in clinical practice, and also research registration and publication, and also dissemination of study results. So it is quite comprehensive. About 10 years after the Declaration of Helsinki was developed in the US, the National Research Act was established, and its main aim was to guarantee stricter regulation and control of clinical trials. In this sense, studies would have to be passed through an institutional review board. Informed consent would have to be obtained from all study participants. Participants would have to be counseled regarding their diagnosis and the corresponding study practices and procedures. And about 5 years after that, finally, the Belmont Report was established, which included the ethical principles and guidelines for the protection of human subjects of research. And it established 3 core principles that should always guide the design and review of clinical trials. And these core principles are the respect for persons, which also has to do with autonomy and the use of consent forms, beneficence, which has to do with the principle of do no harm and to minimize the risks and maximize the benefits for human subjects, and justice, which has to do with avoiding the penalization of human subjects who take part in clinical trials, and also to try to provide a more fair and equitative benefit-risk ratio to the study population. In 1988, the US Food and Drug Administration, the FDA, was given more authority and accountability over the approval of new drugs and treatments. So that was about 32 years ago. And in 1990, the International Conference on Harmonization, the ICH, was assembled to help eliminate differences in drug development requirements for 3 global pharmaceutical markets, namely the EU, European Union, Japan, and the US. And this was aimed at increasing the efficiency in the development of new drugs, and also improving the availability of drugs for the patients and the public as a whole. And finally, in the year 2000, the so-called Common Technical Document was developed. And this document comprised a set of specifications for an application dossier for the registration of medicines designed to be used across Europe, Japan, and the US. So, with this in mind, what are the current trends and challenges regarding clinical research today? We now go on to the brainstorming session, and I have a few questions that I'd like to ask students so we can start a discussion on this topic. So, how is clinical research regulated in your country? Do you know of any scientific misconduct or ethical breaches in clinical research in your country or institution that you would like to bring up for discussion? How do you see the issue of consenting when it comes to children and other vulnerable populations? How to abide by the prevailing ethical standards in the midst of a pandemic such as that of COVID-19? And do you feel that the risk-benefit ratio may be modulated in such circumstances? So these are just a few questions for us to begin with, but you can ask your own questions too, okay? These are the references that I use for this presentation. I'd like to thank you all for your attention. Thanks a lot.